Bright field microscopy. Bright field microscopy is the simplest of all the optical microscopy illumination techniques. Sample illumination is transmitted, i.e., illuminated from below and observed from above white light, and contrast in the sample is caused by attenuation of the transmitted light in dense areas of the sample. Bright field microscopy is the simplest of a range of techniques used for illumination of samples in light microscopes, and its simplicity makes it a popular technique. The typical appearance of a bright field microscopy image is a dark sample on a bright background, hence the name. Light path. The light path of a bright field microscope. The light path therefore consists of a transillumination light source, commonly a halogen lamp in the microscope stand, a condenser lens which focuses light from the light source onto the sample, an objective lens which collects light from the sample and magnifies the image, oculars and or a camera to view the sample image. Bright field microscopy may use critical or Kohler illumination to illuminate the sample. Performance. Bright field microscopy typically has low contrast with most biological samples, as few absorb light to a great extent. Staining is often required to increase contrast, which prevents use on live cells in many situations. Bright field illumination is useful for samples that have an intrinsic color, for example chloroplasts in plant cells. Comparison of transillumination techniques used to generate contrast in a sample of tissue paper 1. 559 mm/pixel. Bright field microscopy is a standard light microscopy technique, and therefore magnification is limited by the resolving power possible with the wavelength of visible light. Advantages. Simplicity of setup with only basic equipment required. Living cells can be seen with bright field microscopes. Limitations. Very low contrast of most biological samples. The practical limit to magnification with a light microscope is around 1300x. Although higher magnifications are possible, it becomes increasingly difficult to maintain image clarity as the magnification increases. Low apparent optical resolution due to the blur of out-of-focus material. Samples that are naturally colorless and transparent cannot be seen well, e.g. many types of mammalian cells. These samples often have to be stained before viewing. Samples that do have their own color can be seen without preparation, e.g. the observation of cytoplasmic streaming in Charas cells. Enhancements. Reducing or increasing the amount of the light source by the iris diaphragm. Use of an oil immersion objective lens and a special immersion oil placed on a glass cover over the specimen. Immersion oil has the same refraction as glass and improves the resolution of the observed specimen. Use of sample staining methods for use in microbiology, such as simple stains methylene blue, safranin, crystal violet and differential stains negative stains, flagellar stains, endospore stains. Use of a colored usually blue or polarizing filter on the light source to highlight features not visible under white light. The use of filters is especially useful with mineral samples.